All right, today we are diving deep into the microscopic world. We're going to become detectives, trying to solve a pretty serious medical mystery. How in the world do you tell a deadly parasite from its harmless twin? So just picture this. You're looking through a microscope and you see these two single-celled organisms that they look almost identical. But the call you make based on some really subtle differences, it could literally be the difference between life and death for your patient. And this isn't just a hypothetical, you know? This is the exact challenge with organisms like Entamoeba histolytica, that's the bad guy, and Entamoeba coli, its harmless cousin. They both hang out in the gut, but one is a destructive invader, and the other, it's just a passenger, not causing any trouble. Getting this right is absolutely critical. So how do we do it? Let's start by opening up our diagnostic tool. Okay, so what's in this toolkit of ours? It's way more than just a fancy microscope. It's all about knowing what to look for, those key clues that make these imposters give themselves away. First up, you've got to know about the trophozoite. You can think of this as the parasite's active duty phase. This is the stage where it's moving around, it's eating, it's multiplying. Basically, it's causing all the problems inside the host's body. And then you have the cyst. This is the parasite in stealth mode. It's like a tiny, tough survival pod. It's dormant, it's got this protective wall, and it can survive out in the world just waiting to infect a new person. This is exactly how the parasite spreads. So, keeping those two forms in mind, the active trophozoite and the dormant cyst, here are the clues we're hunting for. We're going to be looking at everything from the structure of its nucleus and its size to how it moves, and this is a big one, what it's been eating. This is our detective's checklist for nailing the culprit. All right, we've got our toolkit. Let's tackle our first case file, the intestinal amoeba. We're going after the gut invaders. So here we go, a side-by-side comparison of the trophozoites. See, the pathogenic E. histolytica moves with purpose, while E. coli is kind of sluggish, just meandering around. But the real giveaway? It's what's on the menu. E. histolytica is a predator. It's feasting on red blood cells because it's actively invading the intestinal wall. The harmless one, E. coli, it's happy just munching on bacteria. And this is the absolute crucial point. If you see a trophozoid that is actually eaten red blood cells, that's the smoking gun. It is diagnostic for the pathogenic Entamoeba histolytica. Why? Because it proves it's invading the host's tissue. Case closed on that front. Now, what about the dormant cyst stage? The clues here are a little more subtle. I mean, sure, size is one difference, but the real key to cracking this part of the case, it comes down to a really simple counting game. And there it is. Look at that. The pathogenic E. histolytica has exactly 4 nuclei in its mature cyst. The harmless E. coli? It's got 8. It's such a simple difference, right? But for making a diagnosis, it is an absolute game changer. Okay, so we've cracked the case of the intestinal amoeba. Now, let's take these detective skills of ours to a whole new crime scene, the bloodstream. This time, we're hunting down the notorious malaria parasite. Right, so we're talking about malaria, which is caused by the Plasmodium parasite. This thing has a really complex life cycle. It all starts when a mosquito injects the parasite into a person. From there, they make a beeline for the liver, multiply like crazy, and then burst out to infect red blood cells. And that's when all the classic symptoms of the disease really kick in. Now, just like our last case, not all malaria parasites are created equal. The one we're really on the hunt for is Plasmodium falciparum. It's the most dangerous of the bunch by far. To pin it down, we're looking for very specific clues in the blood, things like changes to the red blood cells themselves. And while some of the clues can be a bit tricky to spot, there is one that is just a dead giveaway for P. falciparum. It's this unique signature that no other malaria parasite leaves behind. It's a real telltale sign. And this is it. Seriously, if you remember one thing from this whole section, make it this. Only Plasmodium falciparum produces these unique crescent or banana-shaped gametocytes. If you find these in a patient's blood, that is an immediate major red flag. You know, cracking the case is really only half the battle. The real reason this microscopic detective work matters so much is because the diagnosis you make has a direct line to the treatment and ultimately to the patient's outcome. So when a lab tech spots that trophozoite with the ingested red blood cells, it's a red alert. That confirms an E. histolytica infection, and the clock immediately starts ticking to get the right treatment started. For this kind of symptomatic amoebiasis, The go-to treatment is metronidazole. This drug is designed to specifically target that invasive parasite and stop the tissue destruction in its tracks. An incorrect diagnosis? Well, that might have led to no treatment at all. And spotting those crescent-shaped gametocytes is just as critical. It's the calling card of a P. falciparum infection, which can spiral very quickly into life-threatening complications like cerebral malaria. This diagnosis is the crucial roadmap for treatment. P. falciparum is often resistant to the older drugs, so making the right ID means doctors can go straight for the heavy hitters, like artemether lumefantrine, to save that patient's life. So at the end of the day, telling these microscopic lookalikes apart, it's not just some academic exercise, it's a critical skill. It takes a sharp eye, a deep understanding of these tiny organisms, and it's a skill that directly, without a doubt, saves lives. It really makes you wonder, doesn't it? What other dangerous imposters and harmless doppelgangers are out there, just hiding in plain sight, waiting for some sharp-eyed detective to find them under the microscope?